Okay, I'm Nathan Clark, and I want to say from the very beginning that this work is done in a 50-50 collaboration with my lab as well as the lab of Nathan Morehouse. We are both at the University of Pittsburgh. Let's talk about ejaculates. Ejaculates, of course, are a major, on cue, are a major site of interactions between males and females. This is highly important, right? And ejaculates are, well, they're not homogeneous at all. In fact, very many species produce highly complex and structured ejaculates. We all know of copulatory plugs and spermatophores and such. Well, in our model of choice in butterflies, specifically in Pyrus rapi here, in addition to sperm, males pass to females an enormous amount of nutrition in an internally deposited spermatophore, which in our species can be up to 13% of the male's total body weight. To which the female then replies, "Gee, great, thanks. Now I can go make a lot more eggs." So sexual cooperation in this type of interaction seems to be the goal of this exchange of nutrition, because adult butterflies are extremely starved for nitrogen as well as for energy in general. And we have already shown that this spermatophore contains up to 50% protein by dry weight. And so this is an enormous investment by the males. However, there's kind of a price with this. The females cannot remate, they refuse mates until the spermatophore is digested. So this suggests that there could be a very strong potential for sexual conflict going on in Lepidoptera as well. Because although she cannot remate, she is immediately laying eggs fertilized by that mate's sperm. And so perhaps males that are producing spermatophores that are more recalcitrant to digestion would benefit by delaying mating with other males and avoiding competition in that type of way. For example, in Pyrus rapi, it takes around 2 to 3 days for the female to access the nutrition inside of that spermatophore. And so the female would obviously be very kind of frustrated as to why she can't get to this valuable nutrition, to which the male would just reply, "You need to hold on." Now, how is this achieved physically? The spermatophore can be dissected into 2 discrete regions. You have the interior of the spermatophore, which is rather soft and we would think perhaps easy to digest. Whereas the exterior, we've determined, is almost entirely protein, is extremely tough, and it's completely insoluble, even in strong detergents and reducing agents. The only way we can dissolve it is in hot acid or in very high % bleach. So we think this thing is covalently bound. And so this structure is essentially enforcing a waiting period on the female until she can remate. We've also determined that the protein compositions of the envelope, the outside of the spermatophore, and the interior are vastly different from each other. And so perhaps selection is molding the different parts of the spermatophore for different reasons. We've done intensive biochemical and mass spectrometry work to characterize these. Here's just a taste of it, where we can look at the different proteins from different sections of the male and female reproductive tracts, and determine we've determined essentially that the envelope is assembled from proteins in the more distal, the more proximal portion of the tract, whereas the interior is in the more distal portion. Now, females, of course, are no passive actors in this interaction at all. In fact, they fight back rather strongly. The bursa is completely enveloped in muscle, and it also contains this completely inconspicuous sub-organ you could call the signa, and the signa looks like this. This terrifying structure works to bore a hole through the wall of the spermatophore. In addition to these mechanical ways, we've shown that there are 7 proteases involved and excreted in very high amounts, so that she's also biochemically digesting the spermatophore. This proteolytic activity is seen at very high levels, as measured by Melissa. And so in summary, this spermatophore bursa model is a nice interaction of cooperation and conflict between males and females. And what we'd like to get at very next is what are the consequences of this phenotypically. And we have already some pretty good data showing that there's an incompatibility between distant subspecies, and we can follow this both in vivo and in vitro. Finally, I'd just like to thank everyone. A lot of this work was done by Melissa Plackey, Tamara Sherwood, both in the Morehouse lab, as well as Camille Meslin in my lab. Camille will be leaving for a permanent position in France, so if you would like to be part of this project, please come apply at poster 30 tonight. So thanks for your time.